of all, thank you very much. I was a bit stressed with this connection, I'm not familiar with this platform. So anyway, to start, I have to say that I grew up as a at the university in Collegio Borromeo in Pavia, which is a very old college, but very similar, in fact, to the Sant'Anna. You have to study very hard as in groups, and I think this is one of my best memories which I had in those days, where I really decided to move from being a doctor to start to do research. So I very much regret not having time to spend with you. I would have really enjoyed to visit the Sant'Anna, and I may at some point in the future. I thought today of giving a kind of overview of antibodies, vaccines, maybe also touching a bit on COVID-19, of the new pandemic that actually keeps me here in my office rather than being with you. So to start, I would like to just draw your attention to how much we have gained in life expectancy through a control of infectious diseases. You see what is the life expectancy nowadays in our developed countries, and what was the life expectancy in the Neolithic and Paleolithic as reconstructed from bone remnants. And the reason why there was a very high mortality in childhood and continued over life was actually mainly due to infectious diseases. Now, we know that we are winning, but we have not won completely the fight against infectious diseases with vaccines. And in this table, I want to contrast the fact that we have vaccines against some infection agents. We have eradicated smallpox. We are about to eradicate polio, that until 50 years ago was devastating even in our countries. We can conquer measles and hepatitis B, as well as many other pathogens, but still we don't have vaccines against other viruses like cytomegalovirus, not certainly against HIV. We don't have a good influenza vaccine. And of course, now we badly need a vaccine against SARS coronavirus. Now, how vaccines, I start very low key. Vaccines work by inducing a specific and durable antibody response, okay? You have to boost yourself, but the idea is that once you are vaccinated, the protection can last for many, many years, eventually for a lifetime. And this protection is due to the development of memory, immunological memory, in particular memory B cells that can respond in a secondary antibody response, proliferate and make antibodies in a secondary response, but also long-lived plasma cells that sit in the bone marrow and continuously secrete antibodies that maintain a serum antibody levels. So the serum antibodies provide immediate protection, and memory B cells provide a secondary antibody response. Now, I just want to recall a couple of concepts with this slide. The antibody, how is the antibody combining site made? Is made of 3 loops. If we look at the protein here on the left, you have the, so this is the structure of the V domain. The V domain ends with 6 loops, 3 for the heavy chain and 3 for the light chain. And you see here, if you imagine if you are an engineer, you look into the antibody combining site, you see these 6 loops. And by overlapping different structures, you see that these loops are unique of each antibody. On the right, you see the molecular biology. The antibodies are generated through a fantastic new mechanism of it was new. Susumu Tonegawa, working in our institute, got a Nobel Prize for this discovery. Basically, the idea is that you have only 150 gene segments that in developing B cells are rearranged together in a random fashion with imprecise joining that generate more diversity, and then you pair the VH and the VL, okay? And so in this way, and on top of that, you can introduce somatic mutations. There is an enzyme that introduces mutation in the V region. And these four mechanisms give you an extraordinarily high number of different specificities. There is 10 to the 12 is just one of many estimates. You know, we don't know exactly how many you can make, but this is a huge number. Just think that probably each one of us has only 10 to the 10 B cells. So you cannot even use all this extraordinary antibody repertoire, okay? But keep in mind the 6 loops and the 10 to the 12 specificities. Now, let's go back more than 100 years. Antibodies were one of the first big discoveries and translational discoveries, if you say, made in the early 1900s. In fact, in 1890, Behring and Kitasato started to use horse antisera to protect from diphtheria and tetanus. And this was a breakthrough because until then, people would die of diphtheria and tetanus, and this antisera could be saved. And you see how they were produced. You know, this is not high tech. These are horses which are immunized, and this is a vial of this life-saving drug. Convalescent plasma has been used later to treat severe influenza infection. And you know, even now with SARS coronavirus, you give plasma from convalescent patients. But in recent years, it's possible to make monoclonal antibodies and to use highly potent monoclonal antibodies to treat infected individuals and to prevent the disease, okay? This is called passive vaccination because unlike active vaccination, you just provide passively the antibodies to the individual. Now, the point is that it's difficult to isolate really good monoclonal antibodies by immunizing mice or using other approaches. And so some years ago, we thought that really human memory B cells, which we developed through our lifetime and which remain in our immune system for a lifetime, are really a treasure trove where you can get all these interesting antibodies out. And the reason is that, well, the human immune system is very large, much larger than a mouse immune system. We have 5,000 more cells than mice. There are individual differences in the immune response because of the genetic makeup of each individual, but also because of environmental factors. For instance, we receive stronger stimulation and so on and so forth. People would now say that the microbiota in our gut contribute to the immune responses and so on. But each of us gives a different response even to a vaccine. And then we are repeatedly exposed to human pathogens, for instance, influenza, malaria, herpes. And so we are the best animals to make an immune response against human pathogens because they infect our body and we recognize all the pathogens in action. So to sum up, we have the opportunity from memory B cells to select the most effective antibodies, but also we have the opportunity to study the immune response in real life, in development, in the context not of an animal model, but rather of the response to an infectious pathogen. Okay, so this was almost 10 years ago, we started to develop some simple screening methods, but very efficient. So we decided to either immortalize memory B cells or preserve plasma cells in culture. The reason is that memory B cells can still proliferate, so you can immortalize in clones, while plasma cells are terminally differentiated. So we just keep them alive and screen the antibodies made by memory B cells or plasma cells. And so by screening in a high throughput fashion with a robotized machine, we can have a very highly efficient and high throughput method. We can screen using multiple assays and even functional assays. For instance, we can screen by neutralization. And more importantly, we don't need to know in advance what is the target of the neutralizing antibodies. If you screen by neutralization, you screen for a function, not for a binding to something. And this is important, and we call it a target agnostic approach. And I will explain later with a few examples why it's interesting. Now, the reason to start this was actually a very simple one. It was the time of the SARS coronavirus, and we realized that we could rapidly isolate antibodies from a survivor, from an individual who survived the infection. Okay, and so having learned that, then we isolated antibodies when the MERS coronavirus and other lethal animal coronavirus came up in the 2015. We isolated anti-MERS neutralizing antibodies, anti-Zika antibodies, and at the time of the Ebola, we isolated this very interesting antibody 114 that was shown to potently neutralize the Ebola virus in monkeys, just a single antibody. So here is the idea that zoonotic viruses get into humans. You take individuals who survive, isolate memory B cells, isolate the antibodies, characterize the antibodies, and produce them massively in transfected cells. Now, the first antibody that went into humans is actually our monoclonal antibody 114. And this actually, as a single therapy, can cure Ebola virus infected patients. This has been shown last year when it has been tested compared to other treatments. And if you give the 114 antibody to individuals who are already infected, so the mortality is about 70%, the mortality is decreased to less than 10%. So this is really a life-saving drug that can now be deployed to control Ebola, the spreading of the virus. So in the case of Ebola, here in the title, vaccines or antibodies, I guess the antibodies have the merit that you can use it therapeutically, and you can use it to give it prophylactically to those who have been exposed to the virus. Vaccinating people in the bush would be very difficult, but having an antibody that prevents or at least reduces the mortality is a very useful tool. I have to say that, of course, the antibody was isolated and initially characterized in our lab, but of course has been then developed first at the NIH and then tested in the field. So many different groups have contributed to that. We have just contributed the initial step. Well, let's get to more recent times. SARS coronavirus 2. Well, in this work by the UMAPS and VIR group that was recently published in Nature, we have characterized an antibody called S309. This was isolated from the first individual that survived SARS-1, okay, in 2004. And you see that this antibody has very high affinity for both SARS coronavirus and SARS-CoV-2, exactly the same affinity, neutralizes very well SARS-CoV-2 as well as SARS-CoV-1, and binds to the receptor binding domain of the spike of the coronavirus. You know that this receptor binding domain can be present in a closed and in an open conformation. So the virus hides that face of this domain that binds to the cellular receptor, which is ACE2. And you see here that the antibody can bind all the three RBDs, because in fact this antibody binds on the opposite side of the ACE2. So you see that this is the receptor binding domain, this is our antibody, and this is the cellular receptor of the virus, ACE2. Okay, so this antibody is, we think, is very interesting because it recognizes a very conserved site. It's conserved on 2 different coronaviruses. It's a glycan-containing site. It induces, not only neutralizes the virus, but induces ADCC. So it mediates killing of target of virus-infected cells, and has this unique binding characteristic. Since we have an emergency to face, I think, during this period where everything was closed, our lab, a few people in the lab wanted to help, and so we screened about 5,000 samples from healthcare workers in our region for the presence of antibodies against this domain, the RBD domain, because these are the neutralizing antibodies. And we find a seroprevalence in Ticino, in Switzerland, of about 10%. These individuals make IgG, IgA, and IgM antibodies, sometimes IgG before IgM, which is very counterintuitive, and sometimes actually only IgG. But scaringly, we have seen that several asymptomatic individuals that didn't even know to be infected, they seroconverted, they made antibodies, meaning that have been infected by the virus. And this, of course, is scaring because it means that these individuals, not knowing of being infected, are probably the carriers and the spreaders of the virus. Now, I mentioned the agnostic approach, the fact that we can use antibodies to discover something new. And I want to give you a few examples and tell you how this effort has brought some new concepts in vaccinology to make better vaccines. And the first, so let me go back. The idea here is that you isolate the potent and broadly neutralizing antibodies and use them to identify the target molecule or the target epitope. You can produce the antibody as a drug, but then you can produce the target molecule as a vaccine. And since we are studying this molecule, you can also identify the cellular receptor. Okay, so it's an interesting pipeline that provides you some, as I said, interesting therapeutic tools, but also some interesting tools to study the viral biology. So the first example we stamped in was cytomegalovirus. This is a very complex, very big herpes virus with 20 glycoproteins. And everybody thought that the best target of neutralizing antibodies would be the fusion protein, the G protein. But instead we found that the most potent neutralizing antibodies are anti-pentamer. These antibodies are 1,000-fold more potent than anti-GB. Okay, so these antibodies are now developed as a therapy for severe infections. But more importantly, these antibodies led us to discover this pentamer. So we made this protein, which is actually a pentameric complex of 5 proteins, wasn't easy, and showed that in mice it induces neutralizing titers, which are 300-fold higher than those found in cytomegalovirus infected patients. You see here the results. So with a simple protein vaccine, you can induce a much higher antibody response than the one which is induced by the virus itself. Okay, so this was a very, for us, a very interesting finding, and I hope that this pentamer will be developed in the future. Of course, because we had pentamers and trimers, we made these viral molecules, we were able to identify the receptors for these molecules. One is neuropilin 2, and the trimer receptor is PDGF receptor alpha. And another example are antibodies against the stem of the influenza hemagglutinin. We discovered that these antibodies recognize a very conserved region here in the stem, and which actually paves the way to the development of vaccine just based on the stem, which is conserved, with the idea that they may confer immunity against many different influenza viruses, so a universal influenza vaccine. And another couple of examples. In this study, we identified antibodies against that were very broadly reactive against respiratory syncytial virus, but most importantly, we found that most of the neutralizing antibodies actually recognize the pre-fusion conformation of the fusion protein and not the post-fusion conformation. So there are many antibodies made against the post-fusion conformation, but of course they do not neutralize. So this has paved the way to the concept, which is now well established, that you have to stabilize the pre-fusion conformation in order to make a good vaccine capable of inducing neutralizing antibodies. And finally, just to give you another example, this comes from studies on the malaria, on the antibodies against elicited by the sporozoite, which are the initial parasites injected by the mosquito. We found that the best antibodies actually recognize many different sites, as you see here, NA, NP repeats, but also recognize this junctional epitope, which is not present in the vaccine, which suggests that by adding this extra piece to the vaccine, you may improve the vaccine. So all this to say that being target agnostic, so just focusing on the best neutralizing antibodies and then identify their specificity has been the key to make progress in this field. Okay, I thought to capture your attention, maybe I should go quickly through this slide, because I think at the end what is important in science is to have good questions rather than sometimes more than good answers or some answers. So we have plenty of questions that we would like to address in the field of COVID-19, the disease caused by the new coronavirus. So the first is, which are the target cells of this virus? And is ACE2 the only receptor? So obviously ACE2 is the primary receptor, but there are, are we sure that this is the case? And which are the cells which are infected in vivo? And is the virus cytopathic or not cytopathic? These are questions which are surprisingly still open. Which are the targets of neutralizing antibodies? I talked about the RBD. For sure, the RBD is important. The RBM is this phase of the RBD that really makes contacts with the ACE2, the S1 or the S2 subunits. Now, what is special about this virus is that while antibodies to influenza hemagglutinin, for instance, are always neutralizing, more or less potent, but whenever they bind, they neutralize, there are many antibodies, in contrast, to the spike of the coronavirus that do not neutralize at all. So we want to understand this mechanism. And one of the reasons is that this RBM is kept hidden by the virus. It's only opened up when the spike binds to the receptor. Okay, so there is always a question whether, of course, there are good neutralizing antibodies, whether there are antibodies that do nothing or even enhance either the infection or the disease. This is very important to make sure that with a vaccine we will produce the right antibodies. What's the role of pre-existing immunity against common coronaviruses? Okay, cross-reactive antibodies are probably present in many of us. Do these antibodies protect or do they enhance disease? And then what's the role of innate immunity? It's clear that this virus does not stimulate enough interferon response. What's the role of macrophages, polymorphonuclear cells, and T cells? Of course, again, genetic factors can be important. Age, gender, immune status, microbiota, you name it. And finally, does recovery confer protection? We hear every day that we are not sure about this point. I'm personally convinced that some level of protection is certainly achieved by recovering from this infection. But how long-lived it is, we are not sure yet. and what are the correlates of protection. And all this is important to make an effective vaccine. Okay, so now I want to kind of change gear and move to a more basic questions that we were interested to ask in the context of the regulation of the immune response. And I just want to go back to the concept of the 6 loops and to make the point that the CDR1 and CDR2 are encoded by the V gene, okay? While the CDR3 is encoded by the so-called VDJ junction. So CDR3s are more diverse, but the CDR1 and 2 are encoded by just 50 VH and 50 VL genes, okay? Now, the antibody repertoire is of 2 types. There is a primary repertoire, which is the one generated by VDJ recombination, which is antigen independent. It occurs in the bone marrow before encountering antigen. And then there is a secondary repertoire, which is essentially a fraction of a selection of the primary repertoire of the primary B cells, which are stimulated by antigen and undergo somatic mutations that change the specificity of the variable region and allow the selection of antibodies with increased affinity. Now, in the primary repertoire, the CDR1 and CDR2, as I said, have limited diversity because they are just come from 50 genes. But it's interesting that very often they are polymorphic. Okay, so it means that my V genes are different from your V genes. And having the right V gene is important. This polymorphism impacts on the capacity to respond to certain antigens. We have very interesting examples from influenza and malaria. So V genes matter. The secondary repertoire instead is very diversified because somatic mutations are introduced in all the VDJ sequence. So what's the role of these somatic mutations? What is it? Because there is affinity maturation, because you generate variants which are better binder to the antigen, or just you want to diversify the antibodies, or maybe making an antibody which is more stable, which is a better protein because it's more stable, for instance, to heat denaturation or something like that. Okay, so first example, actually, I want to address this question in the context of influenza neutralizing antibodies. And as an introduction, I just remind you that there are 18 different subtypes of influenza viruses. In human, you have H1 and H3 viruses. They are classified according to the hemagglutinin type. And H1 and H3 are very different from each other. There is only 35% amino acid identity. They are very different proteins. Now, if you look at the classical antibodies that bind here to the globular head of the hemagglutinin and prevent binding to the receptor, these antibodies are very specific for the particular subtype and even for a particular strain, because this region is very variable, okay? But we and others discovered the antibodies that are directed here against the stem of the hemagglutinin and are in fact recognized as sites which are more conserved because these are the sites involved in fusion. This is actually how the fusion occurs, you know? At low pH, the molecules unfolds and springs the fusion peptides into the cell membrane and fuses the two membranes. Okay. So now, how are these antibodies generated? And this is a little bit a busy slide, but I have to say that most of us make these broadly neutralizing antibodies, okay? But only those individuals that have a particular VH169 allelic form with phenylalanine at position 54, okay? 54 position is polymorphic in this VH169 gene and can have phenylalanine of leucine. If somebody is homozygous for leucine, cannot start making this antibody, okay? So the second aspect of this antibody is that there is a tyrosine at position 98 in the CDR3, which is here. Okay? It's frequently found in the CDR3. And then these antibodies can be analyzed by reconstructing the genealogy tree. So basically, let me explain to you. We have isolated all these cells down here. Okay? These are either memory B cells, the elliptic symbols, or plasma cells here. And you look at that these cells carry many mutations. So you see there are, for instance, 27 nucleotide substitution and 15 amino acid substitution going from here to here. But you can reconstruct this genealogy tree, and you discover that the first mutation that occurs here, a proline to alanine substitution at position 52, is enough to get maximum affinity. Okay, so you start with the unmutated antibody that we produce in vitro that has low affinity, and through a single mutation, which is the first to occur, the antibody becomes high affinity. Nonetheless, many mutations accumulate down here. So my question is, why do antibodies continue to mutate when they have already reached very high affinity? Okay. I'm very interested in this question. You may say, well, why do you do that? I think understanding this mechanism of somatic mutation, it's a unique mechanism that the immune system utilizes. I think it's fascinating, at least for me. Okay, this is another example. This is actually a pan-influenza neutralizing antibody. Neutralizes all influenza viruses, again, against the stem. And here we look at the genealogy tree of the heavy and light chain. And what we find is that if you remove all somatic mutations, so if you look at the ancestor of this clone neutralizes very well group 1 viruses, which are in red, but does not neutralize group 2 viruses. So has already very high affinity to start. And through somatic mutation, a variant is generated, FI6, that now neutralizes group 1 as well as group 2. So the mutations now, they have not increased the affinity, they have broadened the specificity of this antibody. Now, what is particularly interesting is that this antibody was isolated from plasmablasts after vaccination of this individual with exactly the same sequence in 2008, 2009, 2010, and 2012. So this means that there is a memory cell in this individual that can be recalled every year and can produce exactly the same antibody without undergoing further somatic mutation. So the model that we come up is that there is priming, there is somatic mutation leading to diversification, and when you boost, you encounter the antigen, you just recall the fittest cells without further somatic mutation. So in other words, you diversify the repertoire within the clone, and then you keep selecting the best cells. I think this is a very, very effective mechanism. I shouldn't say it's a smart mechanism because it's based simply on selection, which is a very powerful but simple approach method. So I think I should finish in the next 10 minutes, but I want to tell you what has been a very surprising finding out of this search for human antibodies. And I have to remind you that malaria parasites is injected by the mosquito. The female mosquito injects 10 to 100 sporozoites. So these are very few parasites. And I think that an antisporozoid vaccine is a wonderful opportunity to provide sterilizing immunity to prevent the infection. But once the parasite gets into the liver and out of the liver in the blood, then you have 10 to the 11 blood stage parasites. So it's a huge load of parasites. Now, these parasites export on the surface of red blood cells, infected red blood cells, the so-called variant surface antigen, which are important to mediate adhesion of the red blood cells of the infected erythrocytes to the endothelia. And it's known that antibodies against VSAs protect from the disease. Now, the problem is that the virus has the parasite, Plasmodia, have more than 200 genes that encodes for these variant antigens. They are extremely polymorphic and they are clonally expressed. So for the parasite, it's very easy to select a different gene and in this way escape the antibody response. So we wanted to find whether there would be antibodies that would recognize all the parasites, okay? So this was again an example of target agnostic approach. We screened the 500 Kenyan plasma to see whether and identified 3 donors that have antibodies in the serum that agglutinate different parasites labeled with different dyes here. And then we isolated antibodies. You see the antibodies listed here. And you see that the antibodies broadly react against these 8 different parasite isolates. You see here 3 antibodies, which are typical antibodies, which are very specific for a single isolate. I told you that these antigens are very, very polymorphic, okay? Instead, we were very pleased to see that these antibodies have a broad spectrum of reactivity against many different parasites. And so we sequenced, and the surprise is that we found that all the broadly reactive antibodies are very unusual. They carry between the V and the DJ a large DNA insertion which encodes a mutated layer 1 exon, and layer 1 is a collagen binding inhibitory receptor which is encoded on chromosome 19. Immunoglobulin genes are chromosome 14. So this piece of DNA comes from another chromosome. It's a templated insertion of DNA into the antibody genes. Okay. Okay, this is the insertion, and this is a crystal structure of the antibody. You know, if you insert a new domain, in this case, in between V and DJ, it ends up on the tip of the CDR3. Okay, now I summarize here quite a few data. So first of all, we identify what these antibodies recognize. It's a class of these variant surface antigens, the refins. The mutated layer 1 domain is sufficient for binding to the refin. The rest of antibody is just a scaffold. So the refin is the binding motif, the binding moiety of these antibodies. In each individual, we isolated many antibodies and many clones. They all have different mutations, but they all have the same origin. So it's a single clone that has expanded enormously, generating different antibodies. And there are many somatic mutations in the layer domain, and it's interesting that some of these mutations abolish collagen binding. Remember, this antibody, if you like, is born as an autoantibody because it binds collagen. But through mutations, you select a variant that does not bind any more collagen. That's good. This is called clonal redemption. And then there are other mutations that modulate the binding to the refins. Okay, good. So how common are these antibodies? We went back to Africa and screened the 2 more cohorts in Tanzania and Mali, and we find that approximately 10% of individuals make these antibodies. And in each case, it's a single clone that gives a very strong antibody response. We find more insertions between V and DJ. So each insertion is a unique somatic event. You see that the piece inserted is always slightly different. And in this case, the domain of layer 1 will be on the tip of the CDR3. But also we find another type of insertion. Here, the layer 1 exon was inserted into the switch region. And because it preserves the splice sites, can be spliced in the mRNA. And now the domain is not exposed on the tip of the CDR3, but rather in what we call the elbow between the VH domain and the CH1 domain. So through splicing, you get this antibody insertion. Are there other examples of these templated inserts? Yes. We now looked whether we could find antibodies that contain LILRB1, which is another inhibitory receptors. Yes, here are 3 examples of 3 African donors. There are large inserts of this LILRB1 in the switch region, which upon appropriate splicing are displayed on the elbow of the antibody. This is a screening of different sera. Now, once again, the refins bind LILRB1. LILRB1 is a receptor that binds class 1 molecules, but the domain 3 is not involved in binding, okay? So the D3 domain can be simply now transplanted by nature on an antibody, and this antibody, without further mutation, binds to the refin and inhibits the binding to LILRB1. And this is a structure of how the refin, you see here, is like an ice cream cone, binds LILRB1. It's like your ice cream bowl. Okay, so at this point we become very intrigued. Let's find whether there are other insertions in the switch region, and we decided either to switch to sequence the switch region or to amplify the switch regions. This is an example of how in an antibody clone with a layer 1 insert diversifies in the repertoire of a single individual, just to give you the impression of how strong is this response. And now looking into the switch region and the VDJ, we find inserts that come from just every chromosome into immunoglobulin genes. They are very abundant. They come primarily from genic regions, introns, and exons, and in some cases, spliceable whole exons, so meaning that they can be expressed. Now, I should wrap up. So, receptor-based antibodies. We find that refins of the malaria bind to a receptor, layer 1 or LILRB1, and the receptor is incorporated in an antibody that then blocks the parasite. Now, this is a completely new model of antibody generation, but actually it's interesting that was proposed 100 years ago by Paul Ehrlich. This is the famous side chain theory. He hypothesized that the pathogen binds to the receptor and the cells react to this by secreting more of this receptor. He called it side chain theory. Paul Ehrlich was wrong in general, but was right in this particular case. Now, because these antibodies use the receptor as for binding to the pathogen, they are unescapable. You know, the pathogen cannot mutate away. And so we think that this explains why these antibodies are so potent, they are so broad, because they use the right element to block the pathogen. And on this ground, we predict that there may be other type of receptor-containing antibodies. And so just to summarize, there are receptors can be incorporated into the immunoglobulin genes, either between VDJ or in the switch region. It's always a single B cell clone, which, however, is expanded and generates a big response. Somatic mutation can still diversify, especially if the insertions in the VDJ, but templated insertions are frequently found in antibody genes of European blood donors. So this mechanism has nothing to do with malaria. Malaria selects the specific cells, but does not induce this insertion. And so, well, we are studying the mechanism at the moment. It's we think that is due to repair of double-strand breaks using patches of DNA that derives from transcribed genes. And in this way, we learned a couple of lessons. We learned how to engineer antibody in the elbow. Nobody expected that it would be possible to insert a new binding moiety in the elbow of an antibody. It works very well. And we also learned new ways to engineer the B cells by offering a substrate that can be inserted when the B cells switch. Now, I don't have, I think I tried to cover perhaps too much ground. We talked about vaccines, how they provide, and antibodies. I can provide active and passive protection, how antibodies can provide a rapid response to emerging viruses. I gave you the example of Ebola. We hope we can do the same with SARS. Antibodies can guide vaccine design. I gave you several examples of how the antibodies can be used to improve, to find the right molecules and to improve the vaccine. I told you about somatic mutations, how they offer the possibility to make variants that can be then selected. And I told you this new story of templated insertion that keeps us very, very busy. I didn't have time to talk about autoantibodies. Now, this is my extended lab a few months ago. Many of my collaborators have now moved to this company, Humabs, that is developing the monoclonal antibody technology, which is based here in Bellinzona. So it's very convenient. They move from the lab to the company. And this is where my new lab will be. I'm moving to the Istituto Nazionale di Genetica Molecolare in Milano, and I'm recruiting PhD students and postdocs. So anybody who is interested to join me to study antibodies, antipathogen antibodies, and mechanism of insertions, please send me an email, and I'll be happy to talk to you. Okay, now with this, I thank you very much for the invitation. I didn't look at the time. Yes, not too bad. And if there are any questions, thank you again. Yes, that's a very interesting question. Actually, we are also working on that. But I didn't have any data to present. Well, first of all, let's start by saying that antibodies are very used to produce. There are, you can produce tons of antibody, literally tons of antibodies in fermenters. So, and injecting antibodies is not a problem. But it's also true that there are ways to let's say, either use RNA or DNA to make the individual cells to produce antibodies. Now, one of the problems is the amount of cells that you need to have in order to reach a particular high level of antibodies in the serum, okay? So these methods are not there yet. Now, you suggest to engineer a B cell that makes an antibody, and this can be done. You can make, there are many ways to do it. But again, in order, this would imply then that you inject these cells, and these cells have to expand and become plasma cells, because the antibody is produced by plasma cells, which have stopped dividing. And so you have to control all these processes. Okay, you see, on the one hand, you have a simple way to use antibodies as drugs. You can inject as much as you want. On the other, if you want to use cells to make antibody, you have several hurdles. Either you inject DNA or RNA and have the antibodies made by, let's say, muscle cells. That's also possible. Or you engineer a B cells and then you transfer these B cells, but then you have to stimulate these B cells to become plasma cells, because only plasma cells make and secrete the antibodies, okay? Many possibilities, and technically feasible. There are always simple way to do things, and I'm not keen of changing as far as they work. So I don't really see the reason to get into complicated genetic manipulation and gene therapy type of approach to make a vaccine, when we can make a protein, mix it with adjuvant, and inject it in a muscle of subcutaneously. Having said that, yes, you can have B cells producing these particular vaccine molecules, but B cells are not very good antigen presenting cells to start an immune response. You can take the RNA or the DNA and just inject it, and that's what many companies are now doing, including for the COVID vaccine. And the proteins are made by different cells and generate an immune response. But yeah, my suggestions would be think carefully because before doing complicated things, if there are simple ways to achieve the same result. And this is a little bit my personal history. All my approach was relatively low key, and I started to use things which are more complicated only where I really needed that. I was never attracted by the technology as such, although we use now a lot of technology. But that's only my suggestion. All what you said is feasible, but do we really need it? I think I'm more impressed by the differences rather than similarity. At least at the structural level and sequence level, there is no similarity. But I'm more impressed by the differences. The first, as I said, for influenza, all the antibodies that bind neutralize. That's not true for SARS. Then some people now speculate that the stem, it's not very clearly a stem, but let's say the stem of the spike protein of the coronavirus may be more conserved, and whether there are antibodies, anti-stem antibodies similar that block the fusion rather than receptor binding, it still has to be proven. We and many others are working exactly to address this question. But this stem, for instance, is very full of glycans, unfortunately. It's very covered with glycans. There is only a little spot which has no glycans and could be a target of the antibodies. I'm also personally not a bit skeptical about the possibility of really making a vaccine just based on the stem of the hemagglutinin. That's where the field is moving now, but I think it's not going to be easy. On COVID, I think the vaccine is going to work. It's very simple. And probably the target of the vaccine is the RBD, this receptor binding domain, certainly the primary target, I would say. So I'm not even sure that you needed to make the whole spike. Maybe the RBD is enough. But you have to cook it in the right way and present it to the immune system in the right way. I'm very optimistic about the vaccine, no doubt about it. But I may be wrong, but I'm optimistic. Let me answer the second part first. No, there is no way that you can guess from a sequence the specificity, unless you have already known that many antibodies with the same sequence have that particular specificity. So at some point, so if you give me an antibody with VH169 and that particular mutations, I will tell you, haha, this antibody may be anti-influenza. But there is no method a priori that you can go from a sequence or a model to the specificity of an antibody. So technically how we do it, yes, for a virus is easy because there are a few suspects. If it is not, you can express the viral glycoproteins in transfect a cell and see whether the antibody binds, okay? Or you may select an escape mutant, for instance, and sequence the mutant. We have done that. For the parasite, for malaria, is much more difficult because malaria has a genome which is almost comparable to the mammalian cell genome. It's a huge genome. Many proteins, I told you. And so in that case, we used proteomics. For instance, you sort the positive and the negative parasites using the antibody. Then you do proteomics and you look at which peptides and therefore which proteins are enriched in the positive and depleted in the negative. And in this way, we have identified the refins and different refins. Then you produce them recombinantly. You order the gene and you check and you show that the antibody really bind to the transfectant. And then you have the specificity. they have a therapeutic effect also alone. That's where I've shown you an example where we actually took just the insert, stick it to an FC, and found and shown that it binds. Okay, so the binding moiety is just the domain which is inserted. Yeah, it is inserted. It's really collinearly synthesized, and it folds an independent domain on the antibody molecule. So what we think happens is that the VDJ recombination breaks the DNA, and in the repair of these breaks, which for antibodies is always made by an error-prone mechanism, this repair is made by, in some cases, by inserting a patch of DNA which comes from somewhere else and has probably been cut out through other mechanisms. We have a hint, it would be a little bit complex to get into these details, but it's essentially an error-prone repair of double-strand breaks. Yes, you could use it. I don't see the. I don't think these antibodies would be terribly useful because I think for malaria it would be much better to interfere with the infection. Okay, so what do you need to fight malaria? Malaria can be cured with drugs. Okay, but you want to decrease the infection so that the parasites doesn't spread anymore. So you need a vaccine that prevents the infection. And once somebody gets infected, takes the drugs. That's really a good question for which I don't have any answer. That's why it's very good. We don't know. It's sometimes it's a bit complicated, isn't it? Microbiota is a very complex topic, and I mean, we would have to start a discussion, an open-ended discussion, and there are it's the mix of the different microbiota that determines an equilibrium. They produce a lot of metabolites, which are very important for the health of the immune system and for the health of the epithelial cells. What's the role of IgA antibodies? Well, I think there is a role, but I can give you an interesting example. One person in 600 completely lacks IgA and is absolutely healthy. Okay. This has been always a mystery. What are IgA good for? Okay. You know that IgA are the main secretory antibody. So it's a complex question that cannot be easily broken down in modules that we can address experimentally. As a pediatrician, I tell you that IgA deficiency is no problem and becomes a problem only if you lack other immunoglobulins as well. But IgA alone, and so in a sense, the answer is that there is no role for IgA. Now, you know that tumors have this neoantigen. Neoantigens are recognized by T cells, and I think it's more a matter, tumors is more a matter of T cells than antibodies, okay? So there are two types of antibodies you can effectively use on tumors. For instance, bispecific antibodies that are anti-tumor, anti-CD3. They make a bridge between killer cells and the tumor so that the killer cells kill the tumor. Or antibodies that recognize antigens on tumor cells that are expressed on tumor cells. But now we are running out of this antigen. Now, the typical is CD19. CD19 is present on lymphomas and leukemias, but also on normal B cells. However, it's true that you can kill the CD19 positive cells, and that's not such a big problem. Of course, it's better to kill all the normal B cells and the tumor than just getting a growing lymphoma. So there are not much anti-tumor antibodies because tumor antigens are either cytosolic protein, which are mutated or just overexpressed by tumor cells, but there are not many examples of surface antigens that can be used as a target on tumors.
Thank you.
There are a few examples, but they're not great examples. And so what we have is the approach of, let's say, killing all B cells and the tumor cells. So it's a non-specific, in a sense. It's not tumor-specific. It's cell lineage-specific approach.